So in addition to the question of the assignment, I add another one that it could be in the test. So these questions are very easy. So first thing that you have to remember, what is a genotype and what is a phenotype. And if you remember the example of Mendel, he was talking about different colors of seeds. So the phenotype will be the appearance, and the genotype will be referred to the genetic endowment of the organism. So first answer will be genotype, and second answer will be the phenotype. And remember that in the case of Mendel, many plants can have the same phenotype. So this tree can have the same phenotype, but the genotype can be different. And here we have in this case, only one option because this one is recessive. So two more questions. So here they're asking you what is the name of, fill this blank about this question, the genotype of an individual that has two different alleles. So there are two different. For a gene is called, so very easy here it will be heterozygous. On the other hand, an individual with the same allele for a gene is called homozygous. So here we see that in this case, this chromosome has the same allele, so this one will be a homozygous, and this one also, although it's recessive, it will be also homozygous. When you have different alleles in the same chromosome, they will be heterozygous. So here they're asking you what is the name of a trait that is expressed in the phenotype when the genotype is either heterozygous or homozygous. So very easy, it will be a dominant. So the dominant, it will be expressed even when you have so for example, this R is expressed when you have two R's, when they are the same letter, or when they are different. So when this one is heterozygous or when this one is homozygous. And on the other hand, a trait that is recessively is only expressed when is, so here is the example, when is homozygous. So this will be the answer. So in this Mendelian principle, we have inheritance patterns of a single, and this is very important, single gene with two alleles. And this principle is called the principle of segregation, and it states that allele pairs separate during gamete formation and randomly unite at fertilization. So, for example, here you see that you have allele pairs in both of them, and they are going to separate each allele during gamete formation, and they will unite randomly at fertilization. So again, you can see it. So here they separate here, and they reunite here during fertilization. And here you find 4 cases where they randomly unite. Okay, so if you go to the Campbell Biology, you will see a long explanation of this in a lot of details, and I highly recommend you that you go and read that in detail. And I just wanted you to remember 3 points from this diagram. First thing, that here we're working with a single gene. And what happened when you work with a single gene? That when Mendel started working with single gene, he noticed a very interesting pattern. When you start working with pure breeds, here, the next generation, you get a hybrid. And this, how you call this hybrid? Monohybrid, because we're only working with one character, with a single gene. And in this case, this monohybrid is a, you see this, is a heterozygous. So this is important, and later when you self-pollinize these plants, so you cross the heterozygous of this generation with the heterozygous, you are going to get this pattern of 3-1 when you have the dominant and the recessive. And as this is called a cross, that is, this type of cross is a monohybrid cross. Okay, so 3 things to remember: that Mendel derived the law of segregation by following a single character, as I show you with this color of the flowers. The second thing that I want that you remember is that the F1 of the monohybrids, there were individuals that they were heterozygous, as I show you here, for this character. And finally, the third one is that the cross between heterozygous is called a monohybrid cross, as I show you here, because we're only talking about one character. Now we're going to move to another Mendel's principle. So in this case, this Mendel principle involved 2 genes. In each one has 2 alleles. So the answer will be independent assortment. And if you see this diagram, that is, I took it from the Campbell biology, that again, I highly recommend you to read it. So here, instead of having 4 options, he has 16 options. And why he has 16 options in this Punnett square? Because his work, he was following 1 trait before, now he's following 2 traits. So in this case, he's following the color of this seed, and also he's following the shape, if it's smooth or if it's rough. So here, so just for you that remember that Mendel identified the second law of inheritance, and how he do it by following 2 characters at the same time. The second thing that he did is he used true breeding prions, and in this case, the 2 characters, they were not called monohybrids; they call it dihybrids in the F1. And why this is important? Because as we will show in the following classes, some traits are transmitted linked or in a package or together, while other ones are transmitted independently. So suppose that we will talk about how those traits are in genes, and if that trait is located, those traits are very far from each other, are in different chromosomes or in different parts of the chromosome that are very far, they can assort independently. However, when you have traits in the chromosome that are very close to each other, so in this part of the chromosome and the other one is very close, they will be transmitted together because they are linked. And this is based on the behavior of the chromosome. And if you have doubts about that, I highly recommend you to go to the Campbell biology, because they explain this inheritance pattern that you have seen multiple times, that is 9331. And what they are trying to do is to show you that there is a connection between the phenotypes and the behavior of the chromosome. That was the next step, to find the mechanisms that explain these patterns of inheritance. So to understand the principle of independent assortment, scientists have to study the process of meiosis. And I know that when you study in high school, you study that when you have these cells, these gametes, they have a first division. Now that you get 2 cells, and then they go to a phase of second division. So they have a second cell division. So now you have 4 options, and this is how gametes form, as we watched from these are the 4 options of the Punnett squares. Yeah, and but when you have when scientists went and study this, as you can see here, so the chromosomes, they found that the chromosomes align in the middle, and here you will see that some, the blue ones will go here, and the pink will go here, so they will go for one cell here, and they will go to another cell here. What is interesting about this process is that you can have this happen randomly, so they don't, it's not that they have a specific chromosome with the dominant that goes, dominance will go here and recessive will go here. No, this can, you can find one gene dominant that can go here, well, and the other gene the recessive and the dominant can go here, or on here you can go, the two dominants can go here. So it's more a random process. So let me see if you explain if this is easier to explain in this way. Here you have the process of meiosis that I showed there. Here you find the first division, and here you find the second division, and here you have the 4 cells that I told you, and here you have other 4 cells, and here we have uh, the different phenotypes that we can get. So here we, in the case that we study, was the color of the seed, and here the shape of the seeds. And what happened here is that, as I said, this could the genes will be located in the chromosome, but they will move randomly. So you have, for example, these heterozygous. So some of them, you have some genes that it will be green for the seed green, and some for the seed yellow. And they, during the division, they will divide. And for the second division, you will have some of them in some chromosome, in some cells, and other ones in other cells. And the same thing will happen for the rounded. So you will have round and wrinkle, and here they will divide, and then they will randomly locate. So you will have some of them that they will be dominant and recessive, some of them dominant and recessive, some of them that they will be both recessive, and some of them that will be both dominant. And again, this happen randomly, and all this mechanism is based on the movement of the chromosomes during meiosis. And again, this is very well explained in the Campbell biology. If you didn't study biology, if this is, I'm not reminding you something that you studied in high school, highly recommended to go and read the Campbell biology. And just this is just a quick reminder. So the law of independent assortment showed that each alleles segregate independently during the gamete formation, and these only apply for genes that are in different chromosomes, genes that are far apart on the chromosome. However, if the genes are located very close from each other, they will be inherited together, so they will be linked. Okay, this exercise is very simple, and I already explained it in the quiz. I think all of you got it, so it's very easy. Now let's move to another one that we haven't studied. Okay, now let's try to solve this question that is also very easy. And here you have to, you start with this is your parental strains that are the homozygous dominant, and here are the homozygous recessive. And you have to get the heterozygous, the proportion of heterozygous in the F2. So it's very easy. So you have to do exactly what Mendel did with the principle of segregation. So you go parental with the pure breeds, and then you get this F1 that will be heterozygous, and then you can get the F2 that that here you have to see the heterozygous. Okay, so let's start doing it. So you have your F1, here you have your parentals, and remember you segregate during gamete formation the two alleles, and then they unite during fertilization, and you get these proportions. All of them are heterozygous, as Mendel showed, and then you have to go and take self-pollinize this. So you will take this, will go here. So you divide, the gametes will segregate again and they reunite randomly, and you get these four options. And if you see here, you will have to be heterozygous, you need to have a combination of alleles that are dominant and recessive, and the answer will be these two. So you will have 2 out of 4, so it will be 50%, or 1 out of 2, so the answer will be E. So it's very easy, and if you have any question, just let me know.